The Great Circle, photosynthesis and cellular respiration. What is photosynthesis? Photosynthesis is the manufacture of sugars with solar energy. The word comes from the Greek photos, meaning light, and synthethonai, meaning putting together. So photosynthesis literally means light putting together. Photosynthesis has two parts. First, light energy will be captured in cellular organelles called chloroplast. Notice that we have all the colors of the rainbow in our arrows. This will occur in leaves. Leaves have structures kind of like solar panels, and the cells of leaves typically contain many chloroplasts. So first, light energy will be captured in these cellular organelles called chloroplast. This captured solar energy is then converted into chemical energy in the form of sugars, which are typically stored in the fruiting bodies of plants. Since the sugars that are manufactured are food, photosynthetic plants and bacteria are called autotrophs, which means self-feeders. Here we have two different autotrophs. One is a plant, Elidea, like the one found in the aquarium in Mr. Hatfield's classroom. The other is a photosynthetic algae, Volvox, which lives in colonies in open water. Both are photosynthetic and both are autotrophs. Now, organisms that cannot make their own food, like animals, are called heterotrophs, which means other feeders. They eat autotrophs or other heterotrophs. Here in this picture, we show some heterotrophs consuming some autotrophs. You should know which is which. Now, autotrophs and heterotrophs typically depend on each other as part of a great circle. In the film The Lion King, Mufasa explains to his son Simba how all living things are connected.
There's more to being king than getting your way all the time.
There's more?
Simba. Everything you see exists together in a delicate balance. As king, you need to understand that balance and respect all the creatures, from the crawling ant to the leaping antelope.
But Dad, don't we eat the antelope?
Yes, Simba, but let me explain. When we die, our bodies become the grass, and the antelope eat the grass. And so, we are all connected in the great circle of life.
Antelopes eat the grass, lions eat the antelopes, but when the lions die, their bodies become food for the grass. And so the circle of life goes on. The Lion King is fiction, of course, and not meant to be taken as literally true. Lions in real life don't have other animals as friends. They have them as dinner. But a great story, even if fictional, can sometimes carry great truths. And the basic idea that all living things participate in a great circle is, in fact, true. Here we see all the colors of the rainbow again. Light energy. The light energy will be captured by producers, photosynthetic autotrophs such as plants. They will use this energy to make sugars for their own bodies, which eventually will be consumed. Consumers are heterotrophs such as animals. They either eat plants or they eat other organisms and thereby obtain energy and raw materials for their own metabolism. Both producers and consumers will die, And as they perish, the remains of their bodies are broken down by other organisms, decomposers, heterotrophic bacteria and fungi. Eventually, of course, all the material is returned back to the trees, back to the plants, back to the producers. They'll use the energy returned to them through the soil, the atmosphere, or the water, and they'll, with another bout of light energy, they'll continue the cycle. The great circle is real. Most of the organisms on Earth participate in the great cycle. So one part of the circle, photosynthesis, occurs in autotrophs. Another part, cellular respiration, occurs in both autotrophs and heterotrophs. Photosynthesis can be summarized with a simple equation. 6 carbon dioxides and 6 waters behind the equal sign are the reactants. And the formula for a simple sugar, glucose, C6H12O6, and 6 oxygen molecules, they are the products. The equation is simple, but the reality behind the equation is more complex. There are actually over 20 separate chemical reactions involved in even the simplest forms of photosynthesis. Students in this course will not be responsible for that level of complexity, but it's well for you to know that there is more complexity available to you. And if you take a college level biology course, you'll get a lot more. You might be responsible for knowing all 20 chemical reactions and their products and even the names of the 20 enzymes involved in that sequence. The equation is simple, but the reality behind the equation is more complex. There are actually over 20 separate chemical reactions involved, and this is just one part of the great circle. To keep things simple, we'll describe photosynthesis as 2 sets of chemical reactions. The first set requires light and are thus called the light reactions. The second set don't require light, and they're often misleadingly referred to as the dark reactions. While the light reactions require light, the dark reactions do not require dark. Thus, the dark reactions can take place at any time with or without light. Because of this, it's more accurate to refer to these 2 sets of reactions as either light-dependent or light-independent. Both sets of reactions in photosynthesis contain steps which are catabolic, breaking bonds, and anabolic, forming bonds. But the net result of all the reactions is an anabolic process that builds sugars out of smaller carbon-based molecules. As mentioned, photosynthesis begins in the chloroplast. Here we see cells of Elidea magnified 100 times. A single one of these rectangular shapes is an individual cell, and the little green dots that we see are chloroplasts. It's interesting to realize that when we look at plant cells under the microscope, as we did in class, that plant cells are actually pretty easy to see. The cells are pretty large, their cell walls are very thick and obvious, and of course, they have structures that are very brightly colored, the chloroplasts, which are very easy to see. One very important pigment is chlorophyll B. Here you see its molecular structure, which has a single metal ion in its center, magnesium. Chlorophyll B strongly reflects green light of a certain frequency, but strongly absorbs other frequencies of light. This illustration showing a background of all the colors of the rainbow is overlapped by a white curve. And you can see that that white line is very low around green and yellow, but it has peaks, one peak between the blue and the violet and the other in the red. So chlorophyll B strongly reflects that green light, but it also strongly absorbs other frequencies of light, as the diagram shows. Again, this illustration shows that all the colors of the rainbow strike a green plant leaf, but perhaps just a little bit of yellow and a little bit of green is reflected and everything else is absorbed. So again, photosynthesis begins in the chloroplast, and it requires chlorophyll and other pigments to absorb solar energy. Chlorophyll B, of course, is green, but it's just one of many pigments found in the chloroplast. Different species may live in environments that receive different colors of light from other plants. For here, example, we have xanthophylls, which are a yellowish-brown color. Here, the picture doesn't show the chloroplast of a green plant, but rather it shows the chloroplast of the giant kelp, a photosynthetic organism that lives in the ocean. Its chloroplasts are yellow. Similarly, other organisms like the rotophyta use pigments like carotenoids, which can range from a light orange to a deep purple. So different living things will live in different environments. They'll experience different colors of light, so they naturally use different pigments to absorb different frequencies of light. And so their chloroplasts may be different colors. Now, to make all this work, chloroplasts have to have a complex internal structure, and we can only see so much of this internal structure with the light microscope. To see more, we'll have to zoom in, and we may have to use other means of resolution. Like other organelles, chloroplasts are formed from molecules called phospholipids. Phospholipids have a property that one side of the molecule is attracted to water and the other side is not. So if you put phospholipids in a watery environment, like the cell, some of the molecules will point to water on the outside and others will point to molecules on the inside. So what happens is the phospholipids form membranes which are double layered. And you're going to see as we dive into the cell more and more, you'll see that almost everything is made of these phospholipid membranes, usually in a double layer. But as you can see here, we have an outer membrane, an inner membrane. Like other organelles, chloroplasts are formed from the folding of double layered phospholipid membranes. Now, these membranes can be folded even more inside the chloroplast. And so we get disc shapes that are made out of folded membrane. We call them membrane-bound disc, and the formal name is a thylakoid. The thylakoid is the actual name for the site of photosynthesis. That's where the photosynthesis is going to occur. So if you think about it, the light has to make its way through the cell wall of the plant, through the cell membrane of the plant, and it has to get through a double-layered membrane for the chloroplast just to get inside to the thylakoid. So that the light has to go through a lot of membranes before it actually reaches the site of photosynthesis. Now the discs are arranged into layers which are stacked. We call these stacks grana, which is plural, the singular is granum. And the interesting thing is these stacked disc-shaped structures, they have a resemblance to electrical transformers, which is very interesting because that's kind of what happens on the surface of the thylakoid membrane. There's going to be a change, and it's going to cause electricity to flow. Now, all this occurs inside a watery environment. The cell is mostly water, and the inside of the chloroplast is also mostly water. So we refer to the fluid-filled inner space inside the chloroplast, this space which surrounds the grana, the stacks of thylakoids, we call that the stroma. So we've got a lot of vocabulary here: outer and inner membrane, thylakoid, grana, and stroma. But the reason why we have to emphasize this is because some of these reactions end up moving positive charge and some of them end up moving negative charge. And these reactions cannot happen in the same place. So you'll be learning that, like when we're moving negatively charged electrons, that's going to happen inside the thylakoid membrane. But when we're accumulating positively charged protons, that's mostly going to happen in the stroma. Now, at this point, you might think we've zoomed in enough and we've had enough vocabulary, but we actually are not seeing photosynthesis yet. We need to zoom in to the very surface of the thylakoid membrane. That means we're going to be zooming in farther than what a light microscope can show you. And this is why we have an electron microscope picture. It's not in color, but then again, these are pictures that are images that are smaller than the colors of light that our eyes are sensitive to. So we'll zoom into these stacks, these grana, and we'll come to an individual thylakoid membrane. Here we will find a photosystem. Photosystems are groups of pigments and other molecules on the surface of the thylakoid. In the illustration, again, you see the stack of the thylakoids. That's a grana. The whole thing's surrounded by the fluid-filled interior, the stroma. But what we're going to do is we're going to zoom in where that green arrow is coming out of. And now you see it, the double-layered phospholipid membrane of the thylakoid. Basically, the thylakoid and the chloroplast and all the other organelles in the cell itself are made of molecules of this type. And here you see the double layer with the heads pointing up, pointing to a watery environment outside the thylakoid, and the heads pointing down to a watery environment inside the thylakoid. Embedded in this membrane on one side is the photosystem. The photosystem is a cluster of photosynthetic pigments. Notice that we have green circles here. Perhaps those green circles could represent chlorophyll B, the most common pigment in green plants. Or here, you see we have different colored discs. A lot of them are green, but we also have some blue and some orange, and those might represent different pigments other than chlorophyll B. Now notice that regardless of what the color is, we see arrows, and the arrows are moving around, and they're moving towards the center of this complex, the antenna complex. We say that photosystems harvest light energy, and we use it to move electrons through the thylakoid membrane. Yes, electrons. The arrows represent electrons being harvested, and they move through the membrane until they come to this central area where there are two embedded chlorophyll A molecules. And so here we have a photosystem, an arrow with all the colors of the rainbow, with all these levels of energy, strikes the membrane, and then we see an electron start to move. In fact, a pair of electrons, 2e minus. And we don't just hit them once, we hit them many times. Again, all the colors of the rainbow strike our photosystem. This causes electrons to move through the thylakoid membrane. We recharge them not once but twice. And the energy of these moving electrons will be used to attach phosphate groups to energy-carrying molecules. Again, light energy strikes the photosystem. This energy is used to move electrons, pairs of electrons, through the thylakoid membrane. So, photosystems use the motion of electrons, 2e minus, and they use them to attach phosphate groups. Notice we have a circle on the bottom of this illustration, capital P, that represents an individual phosphate group. And we're going to attach a phosphate to ADP, and we're going to turn it into ATP. Adenosine triphosphate. This is a common energy-carrying molecule. It's the most common, used by virtually all living things. It's used to store, but also to release energy. It has a three-part structure. We have a double ring structure called adenine. It's a nitrogenous base. That simply means that there's nitrogens attached to the rings. Adenine is one of the 4 nitrogenous bases in DNA, so there's always lots of extra adenine nucleotides inside the cell because the cell always has all the building blocks it needs to make DNA. Attached to adenine, we have a 5-carbon sugar represented by a pentagon. That 5-carbon sugar is called ribose. Ribose is the sugar that's found in RNA. And if you take just one oxygen off that ribose, you turn it into deoxyribose, which is the sugar found in DNA. Attached to this pair, the adenine ribose pair, we have 3 phosphate groups. A phosphate group is simply a phosphorus atom surrounded by some oxygens. And the significance of this is that if you form a bond to attach a phosphate group, that bond is a form of stored chemical energy, which can then be released at a future time to provide energy for some activity of the cell. Now, how is it the cell is able to readily make all this ATP? Well, it's because the molecular building blocks are also used in DNA and RNA, and so they're already present in very vast numbers inside the cell. Here we see the motion of these various monomers. This is just meant to suggest that how they're just floating around, waiting to be used in the fluid-filled interior of the cell. This is very commonplace. Now, it's important to realize that this structure comes in bits and pieces, and the structure has to be built in a series of chemical reactions. So we start off with the nitrogen base, adenine. A bond is formed that attaches it to a sugar like ribose. And if we stop here, this is just called adenosine. It really doesn't have much of a function, but you have to hook these two things together first. Now, if you want to complete it, you can add a phosphate group to the end, which is that phosphate group is useful for collecting, or rather connecting nucleotides of DNA. So we can call this adenylic acid, or more succinctly, we can say AMP, which simply means adenosine monophosphate. By itself, not terribly useful, but this right here, adenosine monophosphate, is the same thing as a single adenine nucleotide of DNA. We'll discuss that more in that unit. Now, if we add an additional phosphate, looking on the first one, we now have adenosine diphosphate. The prefix mono means one, the prefix di means two. And this is a molecule that has a little bit of extra charge that can give it up. But typically, we don't stop here. Our cells, just like plants, are going to make ADP, but then what they'll do is they'll add a third phosphate group. And when we do that, what we get is ATP. Again, making ATP from ADP is pretty simple because these building blocks are already present in large numbers in the cell. To help understand the function of this tiny molecule and why it's so important, we have some analogies. One analogy is that ADP and ATP are like batteries, which are rechargeable. Here we have a picture of a battery, and it has a little graphic on it suggesting it's about halfway full. So ADP, adenosine diphosphate, is like a partially charged battery. But if we add a phosphate to ADP, it is transformed into ATP, and ATP is said to be fully charged. It has enough extra charge stored in it that it can be used to release energy at some future date. Another useful analogy is that ATP is like a coin. It's handy for small amounts of money or energy, but not terribly practical when lots of cash is needed. If one imagines trying to go to a car dealership and buy a new car by paying for it with a big pile of coins, say pennies, you can imagine how inconvenient that would be. On the flip side, we can compare larger carbon-based molecules, sugars like glucose, we can compare them to $1,000 bills of energy. This is certainly convenient for long-term storage. That's why plants make their fruiting bodies and fill them with sugars, right? But while sugars are a good way to store lots of energy long-term, it's not good to try to do reactions with big molecules like this inside your cells. Too much energy would be liberated and your body temperature would go up, and perhaps your cells might actually catch on fire if you did enough of these reactions. So cellular chemistry is typically done with very tiny amounts of energy being stored or released, but the effectiveness can't be doubted. Our bodies, made of trillions of cells, are all doing chemical reactions just like this at the cellular level, and they build ATP and convert it back to ADP on a regular basis every second in order to make that happen.